Now we will start with the glycolysis. Glycolysis, glucose catabolic pathways. There are two phases. One is preparative phase, another is payoff phase. This is the first phase of glycolysis. In this, glucose is converted into a glyceraldehyde. 3-phosphate means 6 carbon converted into a 3 carbons and dihydroxyacetone phosphate. There are first 5 phases, that is called preparative phase, and another 6 to 10 that is called as payoff phase. Okay, this is all about the what is glycolysis. In this, glucose is converted into a glyceraldehyde 3-phosphate. Now, step first, phosphorylations. In phosphorylation, glucose in the cell cytoplasm, glucose 6-phosphate is formed with the enzyme that is called enzyme called hexokinase. Yes, glucose plus hexokinase plus ATP, that will convert into ADP plus glucose and 6-phosphate. And then second reaction, that is isomerization. The enzyme that is phosphoglucoisomerase convert glucose 6-phosphate into isomer, that is fructose 6-phosphate. This isomer have the same molecular formula, but the atoms of each molecules are arranged differently. Here see, glucose 6-phosphate and fructose 6-phosphate, C6H11O6, okay, P1. Same thing is that now glucose plus phosphoglucoisomerase, isomerase is the enzyme that will convert into a glucose into fructose 6-phosphate. Okay, this is then third reaction, that is phosphorylation reactions. Here the enzyme phosphofructokinase use another ATP molecules to transfer a phosphate group to fructose 6-phosphate. Glucose 6-phosphate that will convert into a fructose 1,6-biphosphate. So the reaction will be glucose 6-phosphate plus phosphofructokinase is the enzyme ATP converting into ADP, that is 1-phosphate is utilized by the fructose. So 2-phosphate 1,6-biphosphate is that. Now P2. Okay. Now step 4 is condensation reaction. The enzyme aldolase that split fructose 1,6-biphosphate into a glyceraldehyde 3-phosphate and dihydroxyacetone. Fructose 1,6-biphosphate, or in certain literature you will find diphosphate too. That will convert into a dihydroxyacetone and glyceraldehyde 3-phosphate. That is C3. Okay. C6 convert into a C3. Here is also H5, H5O3, O3, P1, P1. Here is ketose group, aldehyde group, glyceraldehyde 3-phosphate and dihydroxyacetone. Now the isomerase is the enzyme triose phosphate isomerase that rapidly convert into this glyceraldehyde 3-phosphate is removed as soon as it is removed, it used as in next step. So the dihydroxyacetone phosphate, the net result of step 4 and 5, this glucose 1,6-biphosphate convert into a 2 molecules of glyceraldehyde 3-phosphate. Now the substrate level phosphorylation, the enzyme phosphofructokinase transferred a phosphate from 1,3-biphosphoglycerate 2 molecules to ADP to form ATP. Yani 2 ADP is utilized. This happens for each molecules of 1,3-biphosphoglycerate. The process yield 2 3-phosphoglycerate molecules and 2 ATP molecules. You remember 2 molecules of 1,3-diphosphoglycerate plus phosphoglycerate that will convert into a 2 ADP is utilized to produce 2 molecules of ATP. That 2 molecules of 3-phosphoglycerate. Now the step 8, that is dehydration. Intramolecules rearrangement. The enzyme phosphofructomutase relocate the phosphate 3-phosphoglycerate from third reaction carbon to the second carbon to form 2-phosphoglycerate. Here 2 molecules of 3-phosphoglycerate plus, yes, that will convert into a 3-PGA to 2-PGA. Okay, now enolization or enolations. The enolase enzyme remove waters from 2-phosphoglycerate from phosphoenol pyruvate and product. Now end product in the glycolysis PP. This happened for each molecules of 2-phosphoglycerate and 2 molecules of 2-phosphoglycerate. That enolases the enzyme that will gives you a 2 molecules of phosphoenol pyruvate, that is PP. Now the last and final reaction, that is substrate level phosphorylations. In TA cycle we will also understand substrate level phosphorylation here also. Now you remember the enzyme pyruvate kinase transfer the phosphate from PE to ADP2 from pyruvic acid and ATP is formed. So 2 molecules of PEP, that will gives you a 2 molecules of pyruvic acid plus 2 ATP. This is the reaction. Now the whole glycolysis reaction, that is called stoichiometric equations, that glucose plus 2 ADP plus 4 ADP plus 2 NAD plus phosphoric acid H3PO4, that will gives you a 2 pyruvate, 2 NADP and 4 ATPs plus 2 NADP plus H plus and H2O. Okay, this is all about the glycolysis, and these are the steps you will find. First with the glucose will convert into a glucose 6-phosphate, then glucose 6-phosphate convert into a fructose 6-phosphate, fructose 1,6 biphosphate, then 1,3 biphosphoglyceric acid, and then 3-PGA and 3-PGA to 2-PGA, and then lastly convert into a PEP to pyruvate. This is all about the glycolysis. Now the regulation of the glycolysis is include the insulin activate the key glycolytic enzyme, that is glucokinase, phosphofructokinase, pyruvate kinase, and glucocortico inhibits glycolysis. In the summary of years glycolysis, I can say that a single molecules in the glycolysis produce a total 2 molecules of pyruvic acid or 2 pyruvate, 2 ATP, and 2 molecules of NADs and 2 molecules of water. Now glycogenolysis, how it is utilized in the animals, this is not in your course, but to understand these, all the 2 molecules are used in step 1 to 3 and 2 molecules are generated in step 7. 2 more step 10, that gives a 4 ATP molecules produced. If substrate into a 4 minus 2, that will gives you a 2 ATPs used in the step 1, 3. Okay, 4 generated at the end of 10 step, and you end with that is 2 molecules is producing. Okay, so this is all about the glycolysis. Now we will see another chapters.